Yes, a very good morning once again. Derrick Abitegeka is my name, and we are going to continue with our course unit, anatomy and physiology. So far in our past lectures, we have seen the definition of terms that are used in anatomy and physiology, and we have exhausted many of them, and we have looked at other topics such as level of organization. We have looked at abdominal regions, and we have classified them in lectures 1 up to lecture 6. So I urge us to go first to visit those videos in the order so that we can keep together. So after finishing that introductory part of anatomy and physiology, we are going to embark on the topic cytology. And we shall say, by the time we finish this topic, we expect to have seen the definitions under cytology, the types of cells, the structure of cells, functions of the cells, and differentiate between different types of cells. We shall also study the cell division and the cell cycle. So these are going to be our study objectives. And in our previous videos, we have seen that the basic unit, or the smallest unit of life is cell. And we said each living organism exists as long as it has life. It is made up of minute molecules called cells. And we said that there are these cells that are going to come together, one plus the other plus the other plus the other, to form something bigger called a tissue. And when more than one tissues come together, they will form what we call an organ. And in a system where we have more than one organ present, that is called an organ system. And when these organ systems build up together, they form an organism. Just as we saw in our video about levels of organization. So, as I've mentioned them, so it is important for us to begin with that smallest molecule, that smallest unit that is going to build, or that is the active building block for the complex structure called an organism. So, where we study such a small unit is that topic called cytology. In brief, cytology is the study of cells. So here we shall be studying those various cells that make up the body. By the time I say I am an organism, which cells came together to form tissues, to form organs, to form organ systems that today I can exist as an organism. Like we said, anatomy and physiology is a branch of science that deals with the study of body structures in relation to their function. So we shall be studying these cells and relate them to their function, so that we can be able to identify the real complex structure of a human being. And according to objectives, we shall define cytology is the branch of science or branch of biology, because we all know bio means life and logy means study. So we are studying the life. So it's a branch of biology that studies the structure and the chemistry of it. So we study the structure and the chemistry of cells. That's why I say cytology basically will mean the study in the shopkeeper's language. We shall say cytology is the study of cells. But scientifically we are saying cytology is a branch of science or study of biology that studies the structure and chemistry of cells. That is cytology. We shall see a cell. We already defined what is a cell. We said the cell, this is the smallest structural, not that word, and functional unit of life in all living organisms. A cell is the smallest structural and functional unit of life in all living organisms. So, like we said, it is the smallest. Meaning, when these cells come together, they're going to form that complex structure that we are interested in. Then, we shall see what is called the protoplasm. These are some of the definitions we have here. We shall see what we call the protoplasm. And we say, the protoplasm, this is the living structure inside a cell. The living substance inside a cell consisting of cytoplasm and the nucleus. That is what we call protoplasm. So, like I said, cytology is the study of cells. We are studying these cells, and when we talk about the types of cells, we shall see that we have two types of cells. We have what we call the eukaryotic cells, and also have what we call the prokaryotic cells. So basically, since we are doing human biology or human science, we shall deal with eukaryotic cells, because these are the cells we're going to find in human beings. So, as we look at the structure of the cell, we want to appreciate how does a eukaryotic cell look like, how does a prokaryotic cell look like. Like I've said, we have two types of cells. We have eukaryotic cells and prokaryotic cells. So we shall be able to, our main focus is going to be on eukaryotic cells. Though we are going to just have a light about prokaryotic cells so that we can easily distinguish these types of cells. So this is the structure of a eukaryotic cell. Like we have seen, we have eukaryotic cells and prokaryotic cells. So, a eukaryotic cell, this is how it looks like. Or you can make us draw a structure of a eukaryotic cell, or a structure of a human cell, or do the structure of a cell in anatomy and physiology, what they want you to draw is what we call the eukaryotic cell. Why? Because eukaryotic cells are the cells that are found in human beings. Prokaryotic cells are found in other species such as bacteria like E. coli and Staphylococcus aureus cells. You can also find them in archaea, which are most found in extreme environments. So, when they ask you about eukaryotic cell, this is the structure of a eukaryotic cell, and this cell it does not have a specific shape. That is point number one. So, as I've drawn it in an oval form or in a circular form, it does not mean that always it has to be in what? In a circular form. So, it does not have a distinct shape. That is one feature of it. Two, the eukaryotic cell has what we call membrane-bound organelles. So, we have membrane-bound organelles, or organelles within the what? The cell inside. The next feature, it has no cell wall. The eukaryotic cell has no cell wall. It has only one outside membrane, and that is the plasma membrane. Which plasma membrane can also be called the cell membrane. So it is the outside layer of the cell. Another feature is that it has the nucleolus, or it has what we call the DNA. It has what we call the circular DNA. So, like we are going to see, we shall temporize the difference between eukaryotic cells and prokaryotic cells. So, if they ask you with the aid of a label diagram, describe the structure of a eukaryotic cell, describe the structure of a human cell, describe the structure of the cell in anatomy and physiology, this is what they want you to draw. A key emphasis should be put on the label lines. So, the label line should be straight and not crossed at any point. So, we shall describe this structure of a human cell. It has three basic components. The three basic components of this eukaryotic cell. The first component is the plasma membrane. And the plasma membrane is that outside covering. So, the function of the plasma membrane, like we are going to see, like I've said, it has three, let's have it in order. So, we have the plasma membrane, the nucleus, and the cytoplasm. So let us start with the nucleus, and we shall see the nucleus. This is the central organelle. So the word organelle means membrane-bound structures within a eukaryotic cell. So nucleus is an organelle. Golgi body, sorry, mitochondria is an organelle. Golgi apparatus is an organelle. So when we say the nucleus is the central organelle, meaning it is the central one that contains the cell's genetic material and controls the cell activities. In brief, the cell nucleus is the controller of the structure of the cell. And we have said it controls all cell activities, and it also contains the genetic materials of the cell, that is the nucleus. So when you dissect this nucleus inside further, it has what we call the nucleolus. So the nucleolus is inside the nucleus, where we also have what we call the chromatins. The ones that help in DNA building up, or that help in genetic replication of a cell, like we shall see later. So this is the nucleus and the nucleolus. So it is in the nucleus where we have the DNA. That's why I was saying that the nucleus contains the genetic material of the cell, because the nucleus is inside the nucleus. So the nucleus contains the genetic materials and controls the cell activities. Meaning, once the cell nucleus is dead, the cell loses purpose. So that is the nucleus. The second main component I talked about is the plasma membrane. And we shall say the plasma membrane, this is the outer boundary of the cell membrane. So this outer boundary of the cell is what we call the plasma membrane. And one of its functions is to control in and outward movement of materials. Meaning this plasma membrane can allow substances to enter and other substances to move out. So, but that depends on its permeability. So, it being permeable, it means it is impermeable, meaning it can allow substances to go in and others not to go in, depending on particular concentration levels and other factors that we shall see in transport across the cell. So that's the plasma membrane, controls outside and inward movement of materials within the cell, and it also provides shape to the cell. That is the plasma membrane. So, like I said, there are three: the nucleus, the plasma membrane, and the third one is what we call the cytoplasm. So, the cytoplasm, it is the fluid matrix within the cell. So, this fluid that contains or provides movement for all these inner cell organelles is the cytoplasm. And it is a point of chemical reaction in the what? The cell. So that's the cytoplasm. Its function, one, it provides movement of materials within the cell or within the organelles within the cell. This mitochondria can keep moving because of this fluid. So this fluid within this cell is what we call the cytoplasm. The next structure we are going to look at is what we call the mitochondria. So the mitochondria, and we shall say the mitochondria, this is the powerhouse of the cell. The mitochondria is the powerhouse of the cell. When we say powerhouse, it means it is that it generates electricity or energy. So this is how the structure of the mitochondria looks like. So in shape, the mitochondria, it looks like a sausage. So it is in the form of a sausage. For us who know how sausages look like, or that's those of us who enjoy them, it is in the form of a sausage. So its role is to produce energy in the form of ATP. You know very well that the amount of, the body, our bodies need energy to survive at all costs. And what makes this body to keep active, because it uses energy. And that energy that the body needs, or the body mechanisms need to take place, is generated by the mitochondria. And that energy is in the form of ATP. ATP in full stands for adenosine triphosphate. ATP is a form of energy. Meaning, as someone with a lot of energy, someone with a lot of ability to do things, it is because these people have high levels of ATP. And someone who is lethargic or someone who is weak, it is due to low number of ATP. So the presence of mitochondria in the body helps produce ATP that is required for the body to have energy, which energy it uses to carry out all its physiological functions. That is the mitochondria. I think we are together. So this is the mitochondria. Here 1, 2, 3. So you can put any number. That is the mitochondria. The next structure we're going to see is what we call the endoplasmic reticulum. So endoplasmic reticulum, we have two endoplasmic reticulum. We have the one we call the rough endoplasmic reticulum, that is next the nucleus. Then we also have what we call the smooth endoplasmic reticulum. So the endoplasmic reticulum looks like this. If I try to illustrate how it is. So this is how our endoplasmic reticulum looks like. So when it is without ribosomes attached to it, so let me draw them so that you can compare very well. This is another structure of endoplasmic. So we have two endoplasmic reticulum. So this endoplasmic reticulum, we shall attach ribosomes along it. So these structures we have put, I'm going to call ribosomes. So this one is ribosomes attached to it is what we call the rough endoplasmic reticulum. This endoplasmic reticulum without ribosomes is what we call smooth endoplasmic reticulum, because it has no ribosomes attached on its surface. And we shall say endoplasmic reticulum, the rough endoplasmic reticulum, since it has ribosomes, so we have ribosomes attached on it and also have ribosomes floating within the cytoplasm. And the main function of ribosomes is synthesis of protein synthesis. So these ribosomes on the rough endoplasmic reticulum, one, they help in protein and lipid synthesis. So the rough endoplasmic reticulum helps in protein and lipid synthesis, meaning the proteins can be synthesized and can also be stored. So that is the rough endoplasmic reticulum. So it is a network of membranes involved in protein and lipid synthesis. That is the rough endoplasmic reticulum. When you come to the smooth endoplasmic reticulum, for it has no ribosomes attached to it. And the ribosomes, their main function is protein synthesis. So since it has nothing to do with ribosomes, therefore it has nothing to do with the proteins. But its main function is lipid and cellular hormone synthesis. The smooth endoplasmic reticulum helps in synthesis of lipids and steroid hormones. That is the smooth endoplasmic reticulum. It also helps in carbohydrate metabolism and detoxification in the body. So carbohydrate metabolism and detoxification, together with lipid and steroid hormone synthesis, are the functions of the smooth endoplasmic reticulum. I hope we are together. So here we have the rough endoplasmic reticulum, and this is the smooth endoplasmic reticulum as part of the cell organelles. Thank you so much. So the next on our organelle, we are going to see the Golgi apparatus. So the Golgi apparatus, which I have said, this Golgi apparatus, this is how it looks like. So it has coils of cells running from in to outside. So this is our Golgi apparatus or Golgi body. And we shall say the Golgi apparatus is an organelle that modifies, packages, transports proteins and lipids within the cell. That is the Golgi apparatus, and it is this one. So the Golgi apparatus is an organelle that modifies, packages, transports proteins and lipids within the cells. So those lipids and proteins that were synthesized by the rough endoplasmic reticulum and smooth plasmic reticulum will be packed, sorted by and transported to the Golgi body or the Golgi apparatus. That is the Golgi apparatus that we have seen. So then from the Golgi apparatus, we shall move to what we call the lysosome. And the lysosomes, we shall say, the lysosomes are a membrane-bound organelle containing digestive enzymes used to break down waste materials. So when you look at in our structure of the cell, we said this is our structure of the cell. This is the nucleus, and we also talked about the Golgi apparatus, is the cytoplasm as the fluid matrix within the cell. So the lysosomes, we are saying, are membrane bound, meaning they are also found inside the plasma membrane that contain digestive enzymes used to break down waste materials. So for it has what we call digestive enzymes that break down waste materials. That is the lysosomes. Next, we shall talk about the centriole. So the centrioles are cylindrical structures involved in the cell division. Are cylindrical structures that help in cell division. And we all know that cell division is a process how cells reproduce from one level to another. If I can just explain or illustrate how cell division looks, how it happens. So we always have centrioles like this. So if these are my chromosomes, let's say we are at metaphase, and these chromosomes have aligned themselves as spindle fibers. So these cylindrical cells that help attach these spindle fibers. So we have these spindle fibers, these cylindrical structures, and as we are calling them centrioles. So we have those to the left, and those to the right. So these are the centrioles. That's why we are saying centrioles are structures involved in cell division because they help attach spindle fibers that align chromosomes during cell division. That is it. That is the centrioles. Then lastly, let's talk about cell division, but we are going to see in details. This is the process by which cells replicate to form daughter cells. That is cell division. How cells divide from one cell to another. That is cell division. So far we have seen the structure of a eukaryotic cell, the parts that make up the eukaryotic cell, and their functions. So thank you so much for those that are still following us. Before we also finish, we have to see what is a eukaryotic cell. Let us also look at today briefly the prokaryotic cell. But we say prokaryotic cells in human beings are not there. So our main interest will be on eukaryotic cell. But for comparative reasons, and let us see. So we have another type of cell that is called a prokaryotic cell. Like we said, is a single-celled organism that lacks a mature nucleus and membrane-bound organelles. So it is truly the opposite of eukaryotic cell. It lacks membrane-bound organelles, it is single-celled, and it has a true nucleus. We shall say prokaryotic cells are more found in bacteria, more so examples in the E. coli and the Staphylococcus aureus. They are also found in extreme environments like hot springs. Those are the archaea. So that is the prokaryotic cell. So here now we look at the structure of a prokaryotic cell. Like we said, it is single-celled, it lacks a true nucleus, it has a plasma membrane, it has a cell wall, it has a capsule, it has a cytoplasm, it has a linear DNA, it has a flagellum. So this is the structure of how a prokaryotic cell looks like. And unlike we have seen in a eukaryotic cell, this prokaryotic cell has three layers: the outside capsule, the cell wall, the plasma membrane, and the cytoplasm. Like we saw in the eukaryotic cell, it has the nucleate, which is now the genetic material of this prokaryotic cell. The plasma membrane controls in and outside movement of the cell. The cell wall covers the cell components and also controls or prevents loss of external substances, loss of the internal components of this prokaryotic cell to the external environment. It has a nucleate that helps obtain the genetic material of this prokaryotic cell. It also has the ribosomes, which is for protein synthesis. It has inclusions that help in chemical reactions within the cell. It has the pili, what we can call the cilia, that help it to attach to neighboring structures. That is the pili. So this cell can attach to neighboring structures by use of the cilia or the pillars. Then it has the plasmid, and lastly it has the flagellum. So the flagellum, it helps this cell to move. That is the flagellum. So if you have to compare it with the eukaryotic cell, we shall see in eukaryotic cell we didn't have the flagellum, here we have the flagellum. We didn't have the cilia, here we have the cilia. We didn't have the cell membrane, the cell wall, and the capsule. Still, some other organelles, membrane-bound organelles, are missing. We had them in the eukaryotic cell. So next we shall be comparing these various types of cells so that we can basically distinguish, as you are asked to give differences between eukaryotic cells and prokaryotic cells. Thank you so much for watching. Abitegeka Derrick, once again, is my name. God bless you. Let's meet on our next lecture. Thank you. Thank you. Thank you.